Cancer stem cells, or CSCs, have the capacity for long-term self-renewal and the ability to produce differentiated progeny. CSCs have been identified in a growing number of malignancies, including pancreatic adenocarcinoma, in which they can be identified based on aldehyde dehydrogenase activity and expression of cell surface antigens CD44, CD24, and CD133. Here, tumors are isolated from mice harboring human pancreatic cancer xenografts and single-cell suspensions are generated. The cells are then stained with aldofluor reagent, which identifies cells with high aldehyde dehydrogenase activity and cell surface antigens CD44 and CD24. Cancer stem cell populations are then isolated using fluorescence-activated cell sorting. The isolated cells can be used in various downstream functional and analytical assays.
The stem cell hypothesis is an emerging area in cancer biology. In essence, the hypothesis states that tumors contain distinct phenotypic and functional subsets of cells. Certain subsets of cells, termed tumor-initiating cells or cancer stem cells, can generate tumors within immunodeficient mice or colonies within in vitro clonogenic assays. More recently, cancer stem cells are also thought to play an important role in development of metastasis as well as therapeutic resistance. Originally, cancer stem cells were studied in myeloid leukemias using either peripheral blood or bone marrow samples. However, with the emergence of stem cell biology in solid tumors, a challenge has emerged to generate single-cell suspensions that can be flow sorted then studied for downstream applications. My postdoctoral fellow, Zajan Rashid, will now demonstrate this procedure.
I will visually demonstrate several critical steps during this protocol, beginning with the generation of a single suspension of tumor cells from a human pancreatic cancer xenograft. Then I will stain cells for aldehyde dehydrogenase activity and several cell surface antigens. Finally, I will sort cells using a fluorescence-activated cell sorter.
The mice used for this protocol harbor subcutaneous human pancreatic cancer xenografts. These mice were previously generated as follows. Freshly collected tumors from patients that underwent surgery for pancreatic adenocarcinoma at the Johns Hopkins Hospital were minced into 2 to 3 millimeter pieces and implanted subcutaneously into the flanks of 6-week-old athymic mice. When the tumors were approximately 1.5 cubic centimeters, they were excised and transplanted into secondary recipient mice to expand the tumor pool. This protocol begins with the harvesting of a subcutaneous tumor from the flank of an athymic mouse by blunt dissection. Once the tumor has been removed, immediately place it in cell dissociation buffer. Next, weigh the tumor, then place it back into 10 milliliters of cell dissociation buffer per 1 gram of tumor tissue. Working in a sterile biosafety cabinet, transfer the tumor into a 100 by 20 millimeter culture dish containing 1 milliliter of cell dissociation buffer. Using sterile razor blades and forceps, mechanically mince the tumor. Transfer the tumor cell suspension to a 50 milliliter conical tube containing the remaining volume of the cell dissociation buffer. Next, triturate the tumor cell suspension through a 5 milliliter serological pipette 10 times. The tumor pieces should be small so that they do not clog the 5 milliliter serological pipette, and then vortex the suspension at maximum speed for 1 minute. Then, incubate the tumor cell suspension at 37 degrees Celsius for 2 hours, vortexing for 1 minute every 20 minutes. After the 2-hour incubation, vortex the cell suspension at maximum speed for 1 minute. Pass the cell suspension through a 70-micron filter and collect it in a fresh 50 milliliter conical tube. Adjust the final volume to 15 milliliters with DMEM containing 10% FBS and pen strep. To further separate viable cells from dead cells and tissue debris, pipette 15 milliliters of Ficol Pac Plus slowly below the cell suspension to prepare a Ficol gradient for separation by density centrifugation. Take care not to mix the 2 layers. Centrifuge at 500 G for 30 minutes at room temperature with the brake turned off. Following the centrifugation, the viable cells will be at the interface of the Ficol Pac Plus and cell dissociation buffer. Transfer them to a new 50 milliliter conical tube. Add fresh DMEM supplemented with 10% FBS to the cell suspension to dilute it 1 to 3. Centrifuge the isolated cells at 450 G for 10 minutes at room temperature. After centrifuging, resuspend the cell pellet in 1 ml of aldofluor buffer, which is provided in the aldofluor staining kit and will be used in the subsequent cell staining steps. Next, dilute 10 microliters of the cell suspension with 10 ml of trypan blue and count the viable cells using a hemocytometer. If a large number of dead cells or tissue debris are noted during this step, repeat the cell separation by density centrifugation. Cells will be stained for aldehyde dehydrogenase activity using the aldofluor reagent. Cells will also be stained for several cell surface antigens using specific fluorophore conjugated antibodies. Label 7 12 by 75 mm polystyrene test tubes and place them on ice. Dilute the cell suspension in 2 ml of aldofluor buffer to a final concentration of 5 to 10 million cells per milliliter and keep it on ice. Then, according to the instructions in the accompanying written protocol, prepare the following samples to set compensation controls on the fax area flow cytometer. First, non-stained control cells. Second, cells stained with CD44 APC. Third, cells stained with CD24 PE. And fourth, cells stained with mouse CD31 biotin, mouse lineage biotin, mouse H2KD biotin, and streptavidin per CP. Also prepare a fifth sample containing aldofluor only and a sixth sample for setting the ALDH positive and CD44 positive CD24 positive gates. This sample will contain aldofluor, DEAB, an inhibitor of aldehyde dehydrogenase, isotopic control antibodies IgG2b kappa APC, IgG2a kappa PE, mouse CD31 biotin, mouse lineage biotin, mouse H2KD biotin, and streptavidin per CP. Then, in the final tube, stain cells with aldofluor, CD44 APC, CD24 PE, mouse CD31 biotin, mouse lineage biotin, mouse H2KD biotin, and streptavidin per CP. This is the actual specimen to be sorted. Once all of the staining has been performed, keep the tubes on ice in the dark until cell sorting is performed. This sorting protocol will lead to the isolation of ALDH positive, CD44 positive, CD24 positive pancreatic CSCs. Just prior to sorting, add 2 mg per milliliter propydium iodide to the ALDH positive, CD44 positive, CD24 positive gating tube to gate out non-viable cells. Using the non-stained CD44 APC, CD24 PE, mouse lineage, and aldofluor cells, set the compensation controls on a fax area flow cytometer. Generate gates based on forward and side scatter parameters to collect cell singlets. Use the non-stained and mouse lineage tubes to create a gate for non-mouse per CP negative and viable propydium iodide negative cells using the FL3 channel. Most human pancreatic cancer xenografts contain 20 to 60% mouse-derived cells using the mouse CD31, mouse lineage cocktail, and mouse H2KD biotin conjugated antibodies. Using the DEAB treated cells, generate gates in the FL1 channel for ALDH positive cells. The frequency of the ALDH positive population from different xenografts is variable, but is generally 1 to 4% of the total cell population. Then, place the cells for sorting into the fax port and using these gates, collect ALDH positive cells into 12 by 75 mm polystyrene test tubes containing 1 ml of DMEM supplemented with 10% FBS. Next, generate gates created using cells stained with aldofluor, IgG2b kappa APC, and IgG2a kappa PE in the FL4 and FL2 channels. Then, collect CD44 positive, CD24 positive, which generally are 0.2 to 5% of the total population. After each cell population has been sorted, centrifuge the cell suspension at 450 G for 10 minutes and resuspend the cells in 500 ml of fresh DMEM supplemented with 10% FBS. Count the number of sorted cells using a hemocytometer. This procedure will result in the isolation of viable pancreatic CSCs.
These cells can be used for a number of analytical and functional assays requiring viable cells. We've used them to measure in vitro clonogenic growth and invasion. We've used them to measure in vivo tumor initiation and metastasis formation. And we've used them to perform comparative genetic analyses. Once mastered, this technique can be performed in approximately 7 hours. There are several steps that are critical to obtaining a pure population of viable cells. First, xenografts that are less than 1 centimeter in greatest diameter will have the least amount of necrotic tissue within the tumors. This will ensure the least amount of non-viable cells and tissue debris contaminating your final population of isolated cells. Second, while generating a single suspension of tumor cells, it is important to work quickly and maintain the cells or tumor in media supplemented with 10% serum to enhance cell viability. Third, once cell staining has been initiated with the aldofluor reagent and antibodies, it is important to maintain the cells on ice and to keep them in the aldofluor buffer so as to reduce the amount of efflux of the aldofluor reagent from the cells. Additionally, it is important to maintain the cells in the dark as much as possible so as to reduce the amount of photobleaching of the fluorophores. Finally, manually counting the cells using a hemocytometer is important since the flow cytometer will often give inaccurate cell counts.